So, good afternoon, good morning for you. Michael Levin, thanks for being with us. And so the floor is yours for your presentation. You have around 10 minutes, and then we will move to the other speakers and then to the debate. And there will be also plenty of time for you to ask your questions to the speakers.
Well, thank you so much. I'm very happy to be here, and I would like to speak to you today about the collective intelligence of cells as a new roadmap for regenerative medicine. And you can see right away that cells are extremely competent. So this is a unicellular organism. There is no brain, there's no nervous system. All of its needs, the physiological, the anatomical, and the behavioral needs are all handled at the level of one cell. There's no nervous system, there's no cell-to-cell communication, there are no stem cells. Everything is done in this one cell. So single cells are in fact very competent, but the most amazing thing they do is that they work together in groups to achieve very specific goals. So here are some embryonic cells, and they work together to build. This here is a cross-section through a human body. And you can see the amazing, all of the organization of the different tissues and organs that have to be placed exactly in the right orientation, the right size, the right positioning. Everything has to be exactly correct. And so it's very important to figure out where this pattern is stored, because the DNA codes for proteins, so the genome gives you proteins, but what the cells really have to do is build a very complex structure. So we have to understand where the structure is specified. And then we have some animals, such as these are called planaria. These are planarian flatworms. And planaria can be cut into many pieces. The record is something like 275. And every little piece will regenerate exactly what it's missing. So no more, no less, makes exactly what's needed to make a perfect tiny little worm. And so they regenerate their brain, every organ in the body they can regenerate. In fact, planaria are basically immortal. They have no lifespan limit because they continuously regenerate anything that's missing. And even more advanced creatures like salamanders can regenerate their limbs. And so this is what it looks like when you amputate a limb. They will regenerate a perfect copy, and then they stop. And so salamanders can regenerate their eyes, their limbs, their jaws, their portions of the brain, ovaries, spinal cord, incredibly regenerative. So we really have to understand not only how cells build these structures, but how they can repair them and rebuild them after damage. And one of the most interesting things about it is that this process is intelligent, meaning it's flexible and it can make up for novel circumstances. So one example here is a tadpole. So this is a very early frog embryo. You can see the eyes here, the nostrils, and this is the brain. So all of these organs have to move around for a tadpole to become a frog. Okay, so the jaws have to move forward, the eyes have to move, the nostrils have to move. But the amazing thing is that what this creature does is not simply move every piece of the face in some direction, the right amount of distance, but if you make what we call Picasso-like tadpoles, basically these are tadpoles where all the organs start out in the wrong position. So the eyes might be on the back of the head, the jaws are off to the one side, everything is in the wrong position, but they still make pretty normal frogs. So all of these components can move around in novel paths that are not the same as they would normally do in order to achieve a correct frog face. So what the genetics gives you is not a machine that executes specific movements every single time, but actually a kind of error minimization scheme. Okay, it's a system that can handle novelty, like wrong starting positions, and still get to the right position at the end. And this is incredibly important. These responses in that way are flexible. They're not just hardwired. Now, how does this work? Well, remarkably, part of the story is what we call developmental bioelectricity. So it turns out that not only neurons, so this is a familiar story in neurons, that neural cells can make electrical potentials because of these little ion channel proteins, and those potentials propagate to their neighbors, and there's a whole electrical network that can execute intelligence, behavior, and so on. It turns out that all cells do this, okay? And we've created some of the first molecular tools to begin to track the electrical conversations that all of the cells in your body are having with each other. And here you can see, this is a movie made of an early frog embryo, and the different colors are the signal from a voltage-sensitive fluorescent dye. So this is not a model or a simulation, this is a real embryo in time-lapse. And you can see all of the electrical conversations that these cells are having with each other to figure out who's going to be head, tail, left, and right, and so on. And so it's very likely that complex brains evolved by optimizing for speed a very ancient, a very evolutionarily ancient system that was first used by cell collectives to decide what shape they're going to build. Okay, this is the information flow that is used for cells to figure out what they're going to build. And so one application of this is in cancer biology, where when a cell disconnects from the electrical network that tells that what kind of organ to be a part of, they basically revert to their ancient past. They become almost like amoebas, almost a unicellular existence. And so you can see here, even before this tumor is formed, already you can see this aberrant electrical signature of these cells here that are going to disconnect from that electrical network and basically treat the rest of the body as just external environment. And in fact, if you then artificially force these cells into correct electrical patterning, then even though the oncogene right here is very strong, you can see it here, then there will be no tumor, okay? Because it's the electrical connection to the neighbors that makes the cells decide whether to go to metastasis or to build a normal organism. And beyond a single cell control like this, these bioelectric cues have a very important role to play in determining anatomy. So here you see this flatworm again. If you take the middle fragment, okay, so away from the head and tail, you take this middle fragment, it has this really interesting voltage gradient that tells it how many heads it's supposed to have and where the head goes here. And if we manipulate those ion channels and change this electrical pattern, then you can make the cells build whatever you want. And so you can make them build another head, so you get these two-headed worms, or you can get no-headed worms. And the most amazing thing about that is that this electrical circuit has memory. And once you make a two-headed worm, once you amputate, again, this middle fragment, in plain water, no more manipulations of any kind, they continue to form two-headed animals. This change is permanent. So the change to which these cells build after damage is specified by this electric circuit. It's rewritable, it's a kind of memory, and it is not genetic. There are no transgenes here, there's no genomic editing, this is purely bioelectrical. And you can see here in this video what these two-headed animals are like. Not only can you make extra heads, but in fact you can make heads belonging to other species. So in the same triangular-headed species, you can bioelectrically, by manipulating those electrical conversations that these cells have, you can create flat heads like a P. phelina, round heads like an S. mediterranea, or the normal triangular heads. And in fact, their brain shape and the distribution of stem cells will be exactly like these other species. So again, you can shift the pattern to a species about 100 to 150 million years evolutionary distance with no change of the genome. This is the electrical pattern memory that tells these cells what to build. Now, we're starting to use this to repair defects, such as birth defects. So for example, here's a frog brain, here's the normal brain like this. Whether through mutation or exposure to alcohol or various other toxins, you could have these terrible brain defects where the forebrain is basically gone, the midbrain is a big bubble. And so what we did is to build a computer model of the electric circuit that tells the brain how big and what shape it should be during early embryogenesis. And you can ask these models, well, what channels would I manipulate using drugs, for example, ion channel drugs, to create the correct electrical pattern? And when you do that, you rescue the defect. So you get back a normal brain, normal structure, normal IQ, and so on. So this is the beginnings of using computer models to infer bioelectric interventions for regenerative medicine. So in the future, what we would like to do is to have a computer system where you can actually design whatever structure you want. It might be an entirely different animal, it might be a human organ, whatever. And what the system will do is tell you what stimuli, what signals need to be given to those cells to create whatever shape you want. It should be like computer-aided design. And this is critical because if we understand what stimuli will convince this cellular collective to build specific shapes, we would have the solution then to almost all of the problems of medicine. So birth defects, injury, cancer, and aging, it could all be solved if we could control what it is that cells build. And I just want to show you one example of this. And so these are what we call xenobots. They're little proto-organisms made of frog skin. So again, no brain, no nervous system, this is just frog skin. You can see it swimming along here. This is tracking data where many of them are interacting. And you can see here some of them go in circles, some of them can patrol sort of back and forth. And this is one of them traversing a maze. So you can see here that it will, this is still water, there's no water movement. So it will go, take a corner. At this point, for some reason, it decides to turn around and go back where it came from. And so we have a new institute with Josh Bongard's lab at the University of Vermont, which is the Institute for Computationally Designed Organisms. And we want to broadly do two things: make useful synthetic living machines for applications in the body, in the environment, for cleanup, rescue, exploration, and so on, and to use the system to unravel the basic mechanism of cell plasticity. Why and how do cells cooperate to build completely new things? These are made of frog skin, no genetic editing. These are perfectly wild-type frog genome. So I would just like to thank the people who did the work. These are the postdocs and the students who did the work that I showed you. Disclosure of Morphoceuticals, Inc., which funds our work on limb regeneration. And I thank you for listening.
Thank you very much. I think it was really a very inspiring presentation, touching upon many of the, let's say, frontiers aspects of synthetic biology, including this idea of doing computation with living organisms. So this analogy between living organisms and machines or computers or codes. But at the same time, as you have pointed out, it's not exactly like any other machine. It's not that you touch the DNA and you get the results. There are more complex processes like optimization, etc. And then maybe an anecdote that is curious for the people here in Barcelona is that a few years ago I wrote a story on planaria, and I found that many planaria of the Mediterranean variant that are being used in several, or at least were used at that time in several labs around the world, came from Montjuïc, so from a place here in Barcelona where they live naturally. So they are spread all over the Mediterranean, but for historical reasons, many were taken out from here. Okay, so thanks a lot. So now